So let's see. Let's start then. The first talk will be from Mert Basmaci, and the talk is about ontology-based literature mining using the BioBERT model. Hi, Mert. Now it's your call.
Okay. Hello, everybody. And first of all, we would like to thank for Vido's organization for giving us this opportunity to make this presentation. So let me start with the presentation. Here is our outline. It contains introduction, related work, methods and results, conclusion and feature work chapters. So let me start with the introduction part. Protein interactions are vital and play a central role in many biological processes, such as cell metabolism, signal pathways, DNA replication, and more. Further, the disruption of these processes often causes diseases. Therefore, considering their importance, they have been widely investigated in many biomedical studies. Those studies in general have been published and stored in literature databases such as PubMed and Medline. Moreover, there exist several protein interaction-specific databases and interaction networks as well. Those databases and interaction networks store the current knowledge regarding interacting proteins in structural format, and they help scientists to discover the current knowledge regarding the protein pairs. Considering the rapid increase in biomedical literature, manual detection and extraction of PPIs from the long research papers, however, is a time-consuming and exhausting task. Furthermore, updating those databases and interaction networks also sometimes requires manual efforts. Therefore, there is a strong need for the automatic relation extraction techniques and text mining techniques from the biomedical text. So more specifically, here is our problem definition. And our goal is to determine whether pairs of proteins for a given biomedical sentence interact with each other or not. And here we have an example sentence. There we have three protein entities, which are represented by three different colors. And if we check the semantic meaning of the sentence, we can understand that hepatitis B virus X protein is a transcriptional modulator, and it interacts with the transcriptional factor IIB, and it also interacts with the RNA polymerase 2 subunit 5. So there's an interaction between those two protein pairs. And also we have another protein pair, which are transcriptional factor IIB and RNA polymerase 2 subunit 5. But these two protein pairs are not interacting with each other. So our goal here is to detect for both cases. What I mean is we would like to detect both interacting protein pairs, and also we would like to detect non-interacting protein pairs as well. This task in natural language processing, this task is a natural language processing task in computer science, and more specifically, it is a relation extraction task. Previously, many approaches have been proposed for PPI extraction from biomedical sentences. In earlier years, kernel-based techniques have been proposed. Basically, those approaches focus on the spatial sentence representations, such as spatial graphs and tree structures like dependency parse trees, etc. And thanks to the improvements in deep learning domain, recently many deep learning approaches have been also proposed, such as CNN, RNN, or LSTM-based approaches, and they achieved outperforming performance compared to previous studies. So let's move on to the methodology section and introduce how we try to solve this problem. First of all, we have used 5 benchmarking PPI datasets, which are AIMET, BioInfer, HBRD50, IAPA, and LLA. These 5 benchmarking corpora has been transformed into the unified format by PySolo, and they have been accessibly being used by several PPI extraction studies. Therefore, they become a common benchmarking data set by time. And each corpus contains different number of sentences and protein pairs. For example, AIMET and BioInfer are the largest corpora among them, and other 3 corpora is much more smaller compared to these 2. Furthermore, each dataset contains different proportions of positive or negative protein pairs. We can say that AIMET and BioInfer is very imbalanced, considering the positive and negative protein ratio. So it was one of our challenges throughout our study. And for the pre-processing, first of all, we replaced exact protein names with the protein keywords. If a sentence contains a number of N proteins, then there exists and chooses 2 different entity pairs in the sentences, and we evaluate each of these protein pairs separately. First of all, we replaced each protein pairs with protein 1 and protein 2 keywords, and we replace remaining proteins with the protein keywords. And we also have corresponding labels for each protein pairs in our dataset. So we also extend our study by incorporating with INO. Actually, it was enabled by our in-house biomedical literature tool, SciMiner. INO provides a dictionary for the interaction words, and words can be found in this dictionary in several morphological forms. For example, different morphological forms of regulate are regulated, regulates, regulating, regulation, regulator. They all exist in our dictionary. And more specifically, INO is an OBO library ontology, and it reorganizes more than 1,000 interaction terms and their synonyms. For example, the similar words for increase is augment, elevating, rise, enhance, etc. So they all are also specified. Furthermore, INO also specifies the interaction types and hierarchical relations. Here, increase is a type of induction, which is under the positive regulation. And positive regulation belongs interaction type, and interactions are processual entities, and all of them falls into the entity category. So let's return our pre-processing part. As a second setting, we also incorporated with INO, and for this setting, we have also applied a further pre-processing step on top of the first setting. This time, we also tagged interaction words, and we find each interaction word by matching those with the interaction dictionary of INO. And we add special tokens INT surrounding the interaction keywords. So it is our second step, and it is our second pre-processing step. And so we can move on to our learning model, BioBERT. Actually, it is a biomedical language representation model designed for bio-NLP tasks. First of all, we trained 2 separate models for our 2 different settings. In the first setting, we didn't use any ontology, and in the second setting, we incorporated by INO, and we also tagged interaction keywords with special tokens. After those preprocessing steps, we obtained preprocessed sentence, and we give those sentences into our BioBERT tokenizer. Actually, BioBERT tokenizer encodes each of words into the corresponding IDs, and it also adds CLS and separator token to the beginning and to the end of the each sentence. Later, we feed encoded sentences to our BioBERT base layer. BioBERT base is a pre-trained language model which is trained on PubMed abstracts, around 4.5 billion words, and it is also trained with some full PMC text articles with 13 billion words. So we fine-tune this pre-trained base layer with our encoded sentences, so that our final model is capable of predicting protein-protein interactions. So for this purpose, we added an additional output layer on top of the BioBERT base model to enable sentence classification. And as we feed our PPI data during the fine-tuning process, the whole pre-trained BioBERT model and our additional output layer will be updated so that they can perform PPI extraction task. Here are our hyperparameters for fine-tuning the BioBERT model, and here are the results for our two different settings. As an evaluation metric, we have used precision recall and F1 score, and we performed tenfold document level cross-validation, where sentences from the same document can be either in a training dataset or test dataset. For the document splits, we have used exactly the same settings with the IROLA, which allows direct comparison of results. Since each fault yields different precision recall and F1 scores, we calculated their average, and we report the mean of them accordingly. And we only compared our study with those that uses same document level splits and with the IROLA. And the BioBERT model outperformed all of the previous studies on two largest corpora, AIMET and BioInfer, and also on the smallest dataset, LLR. Furthermore, we obtained second best results on the other two datasets. As a second setting, since we tagged each interaction words as well, we obtained a better precision scores compared to first setting on BioInfer and HBRT50 corpora. But this precision improvement is not so clear in this table for the AIMET dataset. So we made a further analysis for the AIMET dataset. And this time we grouped each sentences of AIMET dataset according to the number of interaction words occurrences. The sentences mainly contains 3 or more interaction words. There are around 200 sentences in our datasets for this group. This explains why our overall precision score in AIMET is lower than the expected. More specifically, we still get a higher precision score in the second setting for the first 3 groups, which are no interaction keywords, 1 interaction word, and 2 interaction words. We get still a better precision scores here, but our score dramatically fails if a sentence contains 3 or more interaction words. So I think the model a little bit confused if there are more than 3 interaction words. The reason for this might be there are too many interaction words in the sentence, and very few of them actually indicate the interaction for a protein pair. So as a conclusion, we showed that BioBERT model achieves state-of-the-art F1 scores on 3 out of 5 benchmarking PPI datasets, and obtains the second best result on the other 2 datasets. Also, we incorporated with INO by tagging interaction words and increase the precision of our system. In fact, it may be preferable, especially in the biomedical domain, having a higher precision score. And the incorporation of INO also enabled the additional information regarding protein pairs, such as interaction types. So in the future, we would like to add further ontology support into our study. Since INO provides semantic hierarchies and meanings of interaction keywords and interaction types, INO can be used to help identify potential interaction direction in the future. And we would also like to integrate ontology-based machine learning approaches to support complex interaction mining and analysis. Here is our contact info, and I would like to express my special thanks to my professors, Arzucan, Yunkung, and Oliver. So that's all from my side. If you have any questions, we would like to answer them.
Hey, great talk. Questions? Yes. We will.
Thanks for the great presentation. I'm curious about the situation where the system did not work, but seems to be working pretty well. What was the typical situation of the system cannot be?
What can you hear? Did you hear the question?
I cannot hear it very well. I'm sorry.
Same problem?
Yeah, we had the same problem. Yesterday and today we had this problem. I don't know, it seems like it's only this room. The other rooms are okay. But yeah, seems like he cannot speak. Is it right? Mert, can you hear us?
Yes, I can hear you.
Did you hear the question?
But I couldn't hear the question. I'm sorry.
I mean, the microphone's here, so if you don't talk loud into that to project for that, it's not going to pick it up.
I see.
I'm just going to talk here for a second.
Okay, go ahead.
Okay, so thank you for the presentation. It was really great. Your system seems to pick up most cases, but I was curious, when the system breaks down, what is the limitation? Do you see any pattern on what the system is not able to pick up?
Yeah, especially for our setting, second setting, if we have more than 3 or more interaction words in the sentence, the model dramatically fails to detect interacting protein pairs. When the sentence becomes more complicated, our performance decreases in those cases. But if there are a less number of interaction words in a sentence, let's say 1 or 2, our model is very capable of detecting interacting protein pairs. So actually, we got better results compared to previous studies for those cases.
You wanna? Thank you.
Hi, Mert.
Hello.
Thank you for the presentation. So in biomedical literature, many a times you have gene symbols and protein symbols. I mean, they are the same, right? So in a sentence, you could have verbs which connect both genes and proteins or proteins and proteins. How is it able to distinguish it?
Actually, we didn't distinguish between genes or proteins because actually we replace exact protein or gene names with the protein 1 or protein 2 keywords. Therefore, at the end, our model doesn't know the exact protein names. Instead, it only sees that protein 1 interacts with protein 2, for example. Therefore, we cannot distinguish between genes or proteins. But semantically, our model can understand that semantically there is an interaction between two protein entities rather than their types. But since actually we exactly know protein entities, exact protein entity names, and if we use separate tools such as SciMiner for example, if we use separate tool in order to detect the kind of the protein entities, maybe this tool can help us to state which type of this protein entity. It is a protein or it's a gene. Maybe we can detect it by an additional tagger tool.
Thank you.
Is this answer? Okay.
Okay, that's it. Okay, great. Thank you.